Last video, we talked about understanding the reproducibility crisis and laid out the framework of sensitivity, specificity, and positive and negative predictive value for understanding the reliability of hypothesis tests in which we're declaring statistical significance. And we discovered that, in fact, it's not difficult to actually get to fairly surprisingly low positive predictive value for statistically significant results using very typical assumptions about power and type 1 error rates and prevalence of a true associations among those we're testing. So let's talk a little more about that and what this means for public health. Although we've used the 9.6% prevalence of true associations in the calculations that were examples in the previous slides, I think that actually may be a little optimistic for observational studies. Recall that 9.6% came from clinical trials, which are actually very high stakes, expensive trials to run. And usually a drug doesn't even get to a phase 1 trial unless it's got a lot of promise, and the companies that sponsor those trials really think there's a good chance that it really does have an effect. So I would argue that probably the 9.6% prevalence might be too high for observational studies, which is the kind we usually run in public health, right? We very seldom do formal randomized trials. When we're using observational data, there's sort of a lower cost to accessing data and to going ahead and doing those kind of analyses and those statistical hypotheses tests. And in fact, many of our studies in public health are relatively exploratory studies, by which I mean they're ones in which many risk factors can be investigated at once. And in these studies, at least some, but maybe even most of those risk factors are not actually expected to have true effects. And I think this is increasingly so in recent years, as we now have gotten the ability to measure many things more readily and more cheaply than we have in the past. So, for example, in environmental epidemiology, it's now relatively easy to measure exposure to a number of different chemicals using biomarkers in blood or urine or hair at samples we can get from participants. And we might measure several or even dozens or hundreds of these at a time. It's also now become common to measure thousands or tens of thousands of genes or proteins in these kind of biological samples. And we might look to see if there are associations with many of these genes with a particular health outcome or even with multiple health outcomes. Right? So in these kind of studies, these are really exploratory. We don't have necessarily specific hypotheses going into these studies about which of these genes or which of these biomarkers biologically we think there's a strong rationale for having an association with the outcome. They're really more just generating a lot of data and looking to see if we can learn anything from those. So I'd argue that in these kind of studies, the 9.6% prevalence for true associations is probably way too high. We might expect really more like 1 out of every 100 or 1 out of every 1,000 genes, or maybe even lower proportions to really have true associations with disease. Right? And if we start to plug in those kind of numbers to the positive predictive value formula that we talked about in the last set of slides, what we see is the positive predictive value can drop really quickly. From maybe 64% if we really have a prevalence of about 10%, 1 in 10, to maybe only 14% for a prevalence of 1 in 100, or even 2% for 1 over 1,000. And that's pretty miserable for a positive predictive value, right? If we are down at 1 in 1,000 for our prevalence, then really it would be very rare that a statistically significant finding would actually mean that there's a real association. It's important to understand now, if we think back at our formula for positive predictive value, that it really increases dramatically with adjustment of alpha for multiple comparisons. So this is a technique we haven't talked about yet in this class, but there are methods actually for accounting for when you're doing many hypothesis tests at once. And these are the methods actually that are probably the easiest thing you can do to address the reproducibility crisis as a researcher, as a scientist, is to actually adjust alpha to account for how many tests you're running. Right, the simplest method of doing this, there's actually many different methods have been proposed, including false discovery rate methods, which are become more popular lately. But one of the oldest methods, which is one of the simplest, is called Bonferroni correction, after the person who proposed it. In that correction method, you just take alpha that you normally run, say 0.05, and you divide it by the number of hypothesis tests that you're conducting in any one set of analyses. And if you do this, actually it can bring your positive predictive value way up to reasonable values. So for example, if we have a set of tests now which all have 80% power, and the prevalence of true hypotheses among these tests is 1 over the number of tests, and we use Bonferroni correction, then our positive predictive value actually will always be in the 94 to 97% range, which is actually quite good, right? It tells us that if we get a statistically significant result using Bonferroni correction in this manner, then we're actually got a very high confidence that that actually means there's a true association. Right? And you'll notice here the prevalence is 1 over the number of tests means that, if we basically have a group of hypothesis tests we're running where one of them is actually a true association, then Bonferroni correction will give us a pretty good chance of reliably finding that without getting a lot of false positives. So I mentioned in the last set of slides as well the Unitas paper, which talks about bias, right? Again, these calculations haven't accounted for bias, but in fact, bias is probably occurring to some extent in most fields. And by bias, you can recall, we're not talking about necessarily a researcher that's intentionally biased, but going back to the statistical definition of bias, which we talked about in the first couple of weeks of the course, which is that bias is just the difference between the expected value of the point estimate and its true value. Right, so types of bias that can occur include falsification of results, which hopefully is very rare, although every once in a while somebody does get caught doing this and typically fired from their academic positions or otherwise. But there is actually a lot of unintentional bias that can occur as well. So p-hacking or data dredging, which really just two different terms refer to the same idea, is probably more common, and people do get fired for this as well if they're caught. But this is actually something that we know will lead to bias. And so this basically refers to several different types of behaviors that really have no purpose other than to generate statistical significance in the results. So one thing, for example, that people have done that is p-hacking is to change the outcome or disease definition until they get a definition that gives them statistical significance. So for example, if you had a cutoff on some kind of biological marker that you were going to use to determine whether there's an association with exposure, and then you change that cutoff and try different cutoff values until you see a p-value of less than 0.05, that would be p-hacking. This also includes excluding some observations, and I put serendipitous in quotes here because that's a generous way of viewing this. But again, if you just sort of start to now take out different data points and say, well, is it statistically significant if I exclude person number 1? How about if I exclude number 2? How about if I exclude number 3? And then you finally find some person or some combination of people that would give you p less than 0.05. Again, p-hacking, because you've now just sort of futzed with your analysis for the sole purpose of obtaining a statistically significant result. The simplest way that people do this, and probably the most common and way that people may not even realize is p-hacking, is to keep running statistical models and analyses until you find a statistically significant effect. And probably the most common way this is done is people start to look at subgroups. If they don't see an effect, a statistically significant effect in their main analysis, they'll start looking only at men or only at women or only at young or only at old, and until they find something statistically significant. And again, this is, although there may be some legitimate scientific reasons to do this, for example, men and women are in fact have different biology, and sometimes we've missed effects that occur in one or the other. But if you're doing this only to find statistical significance and kind of repeating this only until you get there, then again, this is kind of, it can be viewed as a type of p-hacking that's really only it's kind of going to lead to bias. And again, the general idea is if you're actually deciding what analyses to do based on whether or not you get statistically significant results on the way, then that falls under data dredging or p-hacking. There's other types of bias that occur, in fact, in many fields, probably including public health. There's some statistical bias from selective reporting of results. This is sometimes referred to as publication bias or the file drawer problem. The idea that, sometimes if you do a simple analysis, you don't see anything, it gets filed away or put in the back of the file drawer because it's not a high priority to publish it. Also, sometimes in some journals, they prefer statistically significant results, and so it's harder to publish. Even if you do try to publish something, it can be harder to get the journal to accept the paper if it's not statistically significant. Some journals are better about this than others. In fact, epidemiology, has been pretty good actually in that field about publishing large studies that are well powered that don't actually show an association. But that's not always true in all fields, in all journals. Another type of selective reporting of results comes from stepwise regression and other types of variable selection, which can inflate the effect sizes and produce bias. Fitting multiple models and reporting the best results, particularly if you define best by which ones give you statistical significance. Again, you're sort of fitting multiple models, but only keeping the ones that are statistically significant and only reporting those. Confounding selection bias or other conceptual errors or limitations that are just inherent in observational study designs can also lead to bias. So how much does bias contribute to false findings? Well, it depends on the proportion of the null effects that are reported as significant due to bias, and this is actually unknowable, right? Because in general, the problem with bias is we don't know exactly what the true effects are. We just get to see what we observe from our imperfect studies. And so in the Unitas study, which I've mentioned before, and I think is available in the supplemental optional readings on the website, that bias parameter U is unknowable, but Unitas proposes that about 10 to 20% might be reasonable for the effect of bias for studies that use standardized methods or a priori statistical models and hypotheses. In other words, the study was planned in advance before looking at any data. All the analyses were specified ahead of time, all the models exactly down to what kind of statistical model and how it would be run and how the data would be treated were designed in advance. And so those would have relatively low bias, he argues, of maybe only 10 or 20%. But more typical epidemiologic studies that include some kind of changing the models or adding hypotheses along the way might be more like 30% for the bias parameter. And then for exploratory analyses that really just don't have any hypotheses and entirely are just based on collecting a lot of data and then dredging through it to see if you can find statistically significant associations, would be more like 80% if you're not adjusting for multiple comparisons, especially in those exercises. Okay, so here's a table from that paper, and you can see here we have several columns. 1 minus beta, of course, is the power. Beta is the type 2 error rate. 1 minus beta is power. And you can see, again, typical high-powered studies, it's got 80, 95% for power and 80%. But it includes some lower power studies with 20% as well. In the Unitas paper, it uses a little different notation, but R is basically just the ratio of true associations to hypotheses, or rather to tests of associations that are not actually there, right? And so, for example, if we've got 1-to-1, that would say that for every true association, there's one test of a risk factor that's not associated with disease. So in our PREV notation for the prevalence, that would be 50% prevalence. And then it goes all the way down to 1 in 1,000 tests actually being true associations. And then U is that bias parameter, again, 10 or 20% for small amounts of bias, up to 80% for large bias. And then he calculates the positive predictive value of the hypotheses tests in those settings and gives some examples of when those might refer to. And again, the idea here is you can just see that it's not difficult to get a positive predictive value below 50%. Right, which is to say, if you hit below 50%, that most statistically significant results are not actually valid, and in fact, many of these examples we get it down to even below 20%, which is pretty bad. Which is the vast majority of statistically significant results are not valid. Okay, so this is fairly well understood by people who are paying attention in science. Now, the question is, what are we doing about it? Well, the NIH, National Institutes of Health, which funds most health research in the U.S., has proposed a solution to this. So there's actually a paper from the head of NIH, Francis Collins, and a co-author discussing initiatives that NIH has explored to restore the self-correcting nature of preclinical research, as they call it. And this is a paper in Nature in 2014 where they laid this out. In that paper, they talk about this growing course of concern, contending that biomedical research is in fact not reproducibility and that our systems are failing and in need of restructuring. And encouragingly, the leaders of NIH said, we share this concern, we explore here some of the significant interventions that we are planning. All right, so the new guidelines they released that started officially in 2016, 2017 required now that research grant applications describe the strengths and weaknesses of prior research being cited by the applicant, and that being crucial in judging the merits of these applications. Scientific rigor, a strict application of scientific method to ensure robust and unbiased experimental design and methodology and reporting results, including full transparency, consideration of relevant biological variables like sex, age, weight, and underlying health conditions, and authentication of key biological and or chemical resources like cell lines, antibodies, other biologics that are used in research. Well, I look at these lists of solutions or proposed solutions to reproducibility, and I have to say I'm pretty worried that this is not going to address some of the key problems. If you recall the positive predictive value framework, although some of these issues may address a few of the bias points raised by Unitas, remember the basic framework for positive predictive value really was dependent only on several characteristics. The prevalence of true associations among those tested, the type 1 error rate is important, and statistical power. And those things are not actually addressed in this set of guidelines from NIH. I think this is actually very concerning because especially there are ongoing trends in health research that are potentially going to make positive predictive value worse. Again, there's sort of this focus now on omics, on measuring a lot of things, on big data. We have now this ability to measure and link data sets of all different types, not just biological measurements, but all kinds of demographic information. We've got large data sets now with many variables we can look at, and I think this will just lead us to do, again, more and more hypothesis tests, more comparisons that are often exploratory with really lower prevalence than we might have had in the past when we were a little more focused on specific hypotheses. These studies now, which measure a lot of things, have become more expensive if we're doing a lot of particularly biological measurements, and that has led to lower sample sizes, which of course, lower sample sizes lead to less statistical power. Our power calculations are becoming more complicated as we deal with these more sophisticated analyses, big data sets, and more sophisticated statistical methods. And so sometimes you see that people will skip these power calculations in their grant applications. And my view is that if you're not doing a power calculation, then you should probably assume that the power is lower than 80 or 90%, although we don't really know. Also, we're paying more attention to potential age and sex differences, again, which could be a good thing if there really exist. However, when we start to do that, we're stratifying now and looking at smaller sample sizes within each of these groups, and that leads to less statistical power, which will reduce our positive predictive value. There are a few bright spots in trends in health research. One is that journals are, I think, now becoming really more aware about the importance of publishing null findings. And so we may have less publication bias going forward than we've had in the past. Also, grant and manuscript reviewers are paying more attention to multiple comparisons and becoming more concerned about the potential reliability of underpowered exploratory analyses. And so that trend could also help as well with ensuring that our funded research actually and our published research is more reliable. So I'm going to kind of try to summarize some take-home messages here, but there's a nice quote from Tale of Two Cities that I think describes well how I feel about doing science these days. And this, of course, is the opening of Tale of Two Cities. It was the best of times, it was the worst of times. It was the age of wisdom, the age of foolishness. It was the epoch of belief, it was the epoch of incredulity. It was the season of light, it was the season of darkness, the spring of hope, the winter of despair. We had everything before us, we had nothing before us. And I think this really captures my feelings, particularly about using big data in science. We have this ability now to measure and link everything. But this is going to create, I think, a lot of false positives if we aren't very careful about how we do the analyses and interpret the results cautiously. So what can we do as researchers? And this is really important, particularly if NIH and funding agencies aren't going to address these issues head on. Well, one thing we can do is increase the prevalence parameter, right? Think back to our positive predictive value formula. This is one of the key factors. If we increase the prevalence of true associations among those we test, then we're going to have more reliable results. And the way we do that is by doing hypothesis-driven research, right? To really focus on things that we think are plausible, that there might be prior evidence either from biological information or other human studies, but focusing on things that we actually have a pretty good reason to think might be important. We can increase statistical power, and this is basically just doing larger sample sizes and or more efficient study designs. And I think there's been, unfortunately, a bad tradition in a lot of sciences where exploratory analyses are sort of given a break on this because they're new, they're novel. We often don't demand statistical power to be high for those. But I think it's especially important when we're doing exploratory analyses to consider the impacts of lower power on their reliability. And if you're going to do them with low power, to be upfront in acknowledging the limitations of that and understand how limited the reliability will be until they're replicated in a second follow-up study that really is hypothesis driven based on that new finding and is well powered for confirming it. We can decrease alpha, and this is actually the easiest thing we can probably do because it costs us nothing to do this as we're doing research. And remember that alpha of 0.05 is arbitrary and it's chosen by the investigator. There is a tradition of using 0.05, but it really is a tradition that was started because Fisher, who was an influential statistician who really first kind of proposed and popularized hypothesis testing, used 0.05 as an example in his book, and that caught on across a number of fields. But it's really not a set number. We can choose to use anything, and there is in fact a growing movement to use a lower value of alpha across the sciences. So there's a paper signed on by I think about 100 researchers that just came out a couple of years ago, suggesting we all just switch to alpha equals 0.005. To make it 0.5% instead of 5%. And if we do that, that would actually raise the positive predictive value across the board, all of the sciences, through just this simple act of using a more strict type 1 error rate limit. Another thing we can do is decrease alpha by using formal adjustment for multiple comparisons. And this is particularly important if we're going to be doing an analysis where we know we're looking at a number of different hypothesis tests of maybe many risk factors that we're measuring. We can decrease bias by avoiding data dredging and p-hacking and by publishing all results, not just preferentially reporting the ones that are statistically significant. We can be more cautious about the interpretation of our finding. I like to say replication before belief. So don't put much weight on a single study that reports an association. See if that can be replicated by another study before you really believe it. And knowing now what you know about positive predictive value, this should make sense to you. Identify exploratory analyses as you're publishing them. Make it clear which analyses were really the ones you planned to do ahead of time and which ones you thought of later, particularly after you started to look at the data and to play with it. And expect low power, prevalence, and positive predictive value for those exploratory analyses, whether they're the ones you were running or whether they're ones you're reading about that others have done. Finally, I'll just end with a call for better science here. A nice quote from Mark Edwards: "Science should be about pursuing the truth and helping people. If you're doing it for any other reason, you really ought to question your motives." And then finally, my quote: "What p-values actually are measuring and why to do power calculations may be the two most important things you can learn about statistics."